H5N1 avian flu spreads on dairy farms. Airborne and wastewater risks. Airborne and wastewater factors in H5N1 spread on dairy farms. The recent decline in H5N1 avian influenza outbreaks on dairy farms across the United States has been notable, yet the complexities of how this virus transmits within these environments remain a topic of active research. New insights from outbreak hotspots in California indicate that both airborne transmission and contaminated wastewater could significantly contribute to the spread of the virus. A team of researchers from Emory University, along with collaborators from California, Colorado, Michigan, and Virginia, has shared their findings from studies conducted on 14 dairy farms in 2 regions of California. These results were published on bioRxiv, a preprint server for scientific research. While the summer months have seen a reduction in reported outbreaks, sporadic detections of H5N1 in dairy cows persist. For instance, the U.S. Department of Agriculture's Animal and Plant Health Inspection Service, APHIS, recently confirmed another case involving a herd in California, bringing the total number of detections since early 2024 to 1,078 across 17 states, with California accounting for 771 of these cases. Infectious particles in the air during milking. Initially, the primary suspects for the spread of H5N1 were contaminated milking equipment and direct contact with infected milk. However, as outbreaks surged in states like Colorado and California, researchers sought a more comprehensive understanding of the virus's transmission pathways, including its occasional transfer to humans. In their latest study, the team conducted thorough sampling of air, wastewater, and milk from the aforementioned dairy farms. During the air sampling phase, the researchers employed 3 distinct air sampling devices, one of which was an open-face polytetrafluoroethylene (PTFE) filter cassette worn on a backpack to simulate exposure for farm workers. They collected aerosols and droplets from the exhaled breath of individual cows and groups of cows, both in milking parlors and housing areas. Out of 71 air samples tested for viral RNA, 6 returned positive results, including 1 from the PTFE filter sampler. Further testing on 9 additional farms, particularly in Southern California, conducted shortly after positive results from milk bulk tanks, revealed H5N1 viral RNA in 21 out of 35 air samples, with infectious virus detected in 4 of those samples. The research team concluded that these findings confirm the presence of H5N1 viral RNA in the air on farms and demonstrate that infectious H5N1 virus is indeed airborne during the milking process. Wastewater as a potential transmission pathway. To investigate the role of wastewater in the transmission of H5N1, the researchers collected samples of reclaimed wastewater from various farms. This included water used for cleaning milking equipment and dairy parlors, as well as samples from drains, sump pumps, fields, and manure lagoons. The researchers found H5N1 viral RNA at every point in the waste stream, including in manure lagoons frequented by migratory birds and in fields where cows graze. Among the samples with high viral loads, 2 wastewater samples contained infectious virus. The researchers emphasize that these results indicate a significant presence of H5N1 in reclaimed wastewater across dairy farms, suggesting it could be another avenue for the virus to spread among cows, to humans, and to nearby domestic animals. Implications for outbreak management and prevention. In additional experiments, whole genome sequencing of selected air and water virus samples revealed amino acid variants in several instances, including some changes in the hemagglutinin segment that did not appear to be consistently maintained. The researchers also noted variations in the virus present on different farms, which could be instrumental in tracking transmission patterns within herds. By sampling milk from individual udders, the team discovered a high prevalence of subclinical H5N1 infections and a varied distribution of infected quarters that remained consistent over time. Their findings suggest that multiple transmission routes for H5N1 likely exist on farms, potentially including aerosols generated during milking and contact with contaminated water. These insights underscore the necessity for implementing a range of mitigation strategies, such as providing respiratory and eye protection for farm workers, disinfecting milking equipment, treating contaminated milk and wastewater to deactivate the virus, and identifying infected cows, even those that do not exhibit clinical symptoms. What do you think? Could the focus on airborne and wastewater transmission change how we approach H5N1 outbreaks in the future? Are there other factors that should be considered in managing these risks? Share your thoughts in the comments. H5N1 bird flu in dairy cows. Low risk to humans, but still a concern. Warning: the next pandemic? Not so fast. Imagine a virus jumping from birds to dairy cows, and then potentially to humans. Sounds like the makings of a pandemic nightmare, right? But here's where it gets reassuring. Scientists from St. Jude Children's Research Hospital have found that H5N1 viruses in dairy cows are more bird-like than human-infecting machines. For now, the lowdown. 3 key takeaways. 1. Pandemic risk, low for now. Research reveals that H5N1 bird flu viruses from dairy cows still closely resemble their avian cousins, lacking the ability to spread efficiently between humans. But don't breathe a sigh of relief just yet. 2. Individual infection, a concern. While airborne transmission is unlikely, direct contact with infected cattle can lead to human infection, posing a serious risk to farm workers and raw milk enthusiasts. Existing treatments, a silver lining. Current avian flu vaccines and antivirals show promise against these cow-derived H5N1 viruses. However, as the virus continues to evolve, the million-dollar question remains: will these treatments stay effective? Unpacking the science. Since 2024, the detection of H5N1 bird flu in dairy cattle has sparked concerns about potential human transmission. To alleviate these fears, St. Jude researchers put a panel of these viruses under the microscope, comparing them to their avian and human flu counterparts. The verdict? These cow viruses retain more molecular and biological features from their bird origins, making them less likely to spread between humans. Expert insights. These flu viruses from cow udders aren't under pressure to mutate for human infection. Yet, notes corresponding author Richard Webby, Ph.D., St. Jude Department of Host Microbe Interactions. The pandemic risk appears low, but direct infection risks for farm workers and others remain high. First author, Tom Fabrizio, Ph.D., adds, "We found cross-protection against these bovine viruses in patients vaccinated against avian influenza, and our results predict antivirals should work effectively against these viruses." For now, the bigger picture. While the risk of pandemic spread may be low, these viruses have already caused at least 41 human infections through close contact with dairy cattle. As the virus continues to evolve, we must ask ourselves, are we doing enough to protect farm workers and those consuming raw milk? How will we stay ahead of potential mutations that could increase human transmission? Should we reevaluate our approach to avian flu vaccination and treatment in light of these findings? Share your thoughts. Do you think the risks associated with H5N1 in dairy cows are being adequately addressed? Should we prioritize vaccine development for farm workers and raw milk consumers? Join the conversation in the comments below.